Right. Good afternoon. My name is Dan Shelley. I'm the Chief Business Development Officer for PolTREG. PolTREG is a company based in Gdansk, Poland. I am not Polish. I'm based in the U.S. So let me introduce the company and walk you through what we're doing. We're sort of in that niche space in autoimmune. We are developing actually T regulatory cell therapies for the autoimmune space. We're not repurposing CAR T. So I think a couple things that really differentiate us. We are the pioneer in T regulatory cell therapies. We are the first organization to take Tregs into humans in the clinic. We've generated clinical proof of concept in type 1 diabetes and MS. We've built a fully operational cGMP facility in Gdansk, and I'll show you pictures of that in a minute. And in addition to the polyclonal Tregs that have been in the clinic now actually up to 10 years, we have a pipeline of proprietary antigen-specific CAR and TCR Treg programs. This is our disclosure statement. Skip that. So, we were excited when ARM put out a report on the top, I guess there's 12 of them, disruptive technologies in the cell and gene therapy space. And of course, number one is engineered T cells for autoimmunity. We've heard that 2024 is the year of autoimmunity, so we're pretty excited for that. In the report, it also does mention polyclonal Tregs. There's, I think, a lot of excitement for engineered Tregs, but of course, those are probably 5 to 8 years away from approval. And the polyclonal Tregs, you'll see in a minute, are much closer. So I'm sure most of you are familiar with the concept of autoimmune disease, right? This is a disease where the body generates hyperactive T cells and autoantibodies that attack healthy tissue. And Tregs are sort of the policemen that are there to kind of restore balance. They act through a number of different mechanisms. They can directly interact with antigen-presenting cells and prevent the stimulation of T effectors. They can actually secrete cytokines that directly inhibit T effectors. In some cases, they can actually cause cytolysis and induce apoptosis in some T effectors. They can compete for cytokines that activate T effectors, prevent basically them from seeing the cytokines, and they can actually disrupt or prevent proliferation of T effectors, and in some cases can actually stimulate FOXP3 expression in naive cells to produce even more policemen, if you will, Tregs. So what I've done here is we've kind of mapped the autoimmune cell therapy space. On the left side, you see companies playing with some unique cell types, not T cell, but like maybe NK. In the center, that large red box are companies that are repurposing CD19, CD20, BCMA, CAR Ts into the autoimmune space, basically B cell depletion and resetting the B cell compartment. We think this is an interesting approach. We don't know sort of the long-term durability of that approach. And I'll get into in a minute that there could actually be a rationale to combine B-cell depletion with long-term autoimmune suppression with Tregs, so combining CAR T and CAR Tregs. On the right side, you see 4 modalities associated with Tregs. There's the CAR Treg space. Upper right would be polyclonal Tregs. On the bottom, the TCR Treg space, and then the bottom right is a novel modality I'll describe in a few minutes. We call antigen-specific or educated Tregs, and I'll note that PolTREG is the only company that is active in all 4 of the Treg modalities. So, as I mentioned before, we do have clinical proof of concept. We've in fact treated now over 100 patients with polyclonal Tregs. There's a perception, I think, in the industry that polyclonal is an academic exercise, that it's not as exciting or sexy as the CAR Tregs, but I should also point out that polyclonal Tregs can be deployed in any of the 80 autoimmune diseases that are available. If you have a CAR or a TCR Treg, you need a new one for each new indication that you go after. So there's a scalability issue that we think we actually overcome. We've also heard that maybe there's not a market for a polyclonal Treg. And I should point out that of the 100 patients that have been treated, we actually have a hospital exemption in Poland to allow patients to access this therapy when it's not been approved. 30 of the 100 patients have been dosed through the hospital exemption. I should also point out that in Poland, this therapy is not covered under insurance. So these patients are paying out of pocket for a non-approved cell therapy because they actually understand and appreciate the burden of T1D. We've used the exact same manufacturing process, effectively the same cell, just from different people, to treat MS. We'll go through that data in a minute. That's a relatively small phase 1 study with 14 patients, but it was quite promising. At the bottom, you can see we have CAR Tregs for neuroinflammatory diseases. We have, for type 1 diabetes, we have PTG-020, which is this antigen-specific cell I'll describe. And we are actually interested in moving Tregs earlier in type 1 diabetes, and you'll see why in just a minute. So most autoimmune diseases are similar to oncology in the sense that the longer you wait, right, the harder it is to treat. In oncology, it's tumor burden. In autoimmune, it's depletion of, in this particular case, in type 1 diabetes, it's depletion of the beta cells in the pancreas that secrete insulin. Patients are not symptomatic until they're in phase 3, and by that time, they've lost over 70% of their beta cells and their capacity to produce insulin. This is where our first set of data, you'll see in a minute, was produced. Our phase 1 and phase 2 are in early onset stage 3 diabetes. Now, interestingly enough, here in Italy and in Poland, they are starting to screen children, primary and secondary school children, for autoantibodies for type 1 diabetes. With 1 or 2 autoantibodies, you've got about a 30% chance that you're going to develop type 1D. But with 4 to 5 autoantibodies, it's almost a 95% chance that you will develop type 1 diabetes, and therefore it is predictive. And you also notice that as you, okay, the laser pointing doesn't work. So sort of on the left side, you can see that the beta cell mass is preserved. So if we could catch those patients where they still have 90% or more of their beta cells, we might be able to prevent disease. And why do I say that? So, I'll skip this. We now are looking at, in 2024, we're looking at 10-year follow-up data in some of these patients. But the first set of experiments, we looked at these patients 2 years after treatment. So what this experiment is, it's called a mixed meal tolerance test. The patient fasts overnight. They take a boost drink, like a breakfast shake. And you can measure C-reactive peptide, which is a surrogate for insulin production. You can see that the patients that were treated with Tregs still have the ability to secrete their own insulin, whereas the patients that did not receive treatment, all their beta cells are gone now. They've lost the ability to secrete insulin. And I should point out that this isn't sufficient, right? They only had 30% of the beta cells at this time, so they're secreting insulin at about 30% capacity of a normal, healthy individual. But we also see at 5 years that these patients are still able to produce their own insulin. So we've effectively frozen the depletion of the beta cells, which is why I was saying if we can move earlier into the pre-symptomatic patients, this could be theoretically curative. That was our phase 1 study. Our phase 2 study came along, and we decided to include rituximab. This is where I was talking about the combination of a CAR T and a Treg. What you can see is that not all patients do respond, so we do lose some of them, about 50%. With rituximab, we maintain these patients longer, about 18 months of the 2-year treatment, but ultimately the Tregs do quite well. We think part of the problem is rituximab it has limited utility in depleting B cells, where this is why like a CD19 or a 20 BCMA could actually be very interesting to look at. Despite the fact that we get about the same level of patients who still can secrete their own insulin, the rituximab arm does seem to show that the patients can secrete more. So there's a logic to having a combination therapy. So over 2 years, those patients secrete more insulin than the Tregs alone. So I mentioned that we then took the same manufacturing process and the same cell type, and we deployed it in MS. This is relapse-remitting MS. Interestingly, we don't need as many cells, 40 million intravenous and only 1 million cells given intrathecally, which is basically a highway to the brain. And what we can see is on the left, you see something called the Expanded Disability Status Score. This is a scale from 1 to 10. From 0 to 2, these patients have high mobility. They're relatively normal. When you get to 8 to 10, you're in a wheelchair. You might have lost all mobility, and if it's a 10, those patients typically pass away. And what you can see is that both the intravenous and intrathecal over the course of a year prevent progression in MS. But intrathecal has much more tight control in these patients. So we think this is a promising way to go. On the right side, what you're looking at is MRI scans of the plaques that are developed in MS patients in the brain, both the size and the number. And again, you can see intravenous, we do see some progression. In other words, we see more plaques and them getting larger, but the intrathecal approach maintains, keeps those patients from progressing and keeps it tighter control. So we're really encouraged by this, and we'll be opening up 2 phase 2 studies, one for relapse remitting, one for primary progressive towards the end of this year. Now, I mentioned that we are developing a number of novel modalities. Again, in type 1 diabetes, we have the TCR Tregs, and we have the antigen-specific. And then for multiple sclerosis and neuroinflammation, we have the CAR Tregs. So the antigen-specific technology is quite interesting. What we did is we identified patients who were good responders from the polyclonal studies that we did. We were able to go back to those patient original blood samples and identify insulin-specific peptides or insulin-like peptides that were associated with good clinical outcome. We then take Tregs from those patients, we take monocytes from the patient, and we load the monocytes with these insulin-specific peptides. We co-culture the monocytes with the patient-derived Tregs, and those Tregs have endogenous TCRs. They pick up the presentation of these epitopes on the MHC of the monocyte. When that interaction occurs, those T cells, those Tregs proliferate. What we're doing right now is we can dye, using a fluorescent dye, we can add a dye to the Tregs, and those that do not proliferate will maintain a high degree of staining. The ones that do proliferate have been trained against those specific peptides, right? And they'll lose dye every doubling. So we can go through and identify the Tregs that have the least amount of stain, and we can pull them out and then amplify them just like we did, expand them just like we do with the core product. And now we have a population of Tregs that are specifically trained against the appropriate autoantigen. Now and what you can see here on the left is the number of clones that the polyclonal Tregs hit is vast, right? So they are non-specific, they go after a lot of clones. But the group that we can pull out, they are very specific to a very limited number of clones. And so we think that this will be a more effective way, sort of a follow-on, if you will, to the polyclonal Tregs. And then again, we can expand them. Now, that interaction I described for you, where we have the TCRs interacting with these insulin-specific peptides, we can actually identify those TCRs, we can sequence them, and we obtained a European Union grant, 7 million euro, to now generate TCR Tregs based on the antigen-specific technology that you saw. And we are looking at this, again, to do in MS and other clinical trials that we might open up. This is very early stage. We're just awarded this. We've just sequenced the TCRs, so we're in the process of advancing this. And then finally, in a collaboration with a partner, AZ Therapies, we're developing both an autologous and an allogeneic by knocking out the HLA CAR Treg. In the Treg world, CARs are typically designed for localization, right? They tell the Tregs where to go, whereas the TCR Tregs are against specific targets per se, autoantigens that we want to go after and suppress. Right. And what you can see here on the right side, the green cells are the Tregs. The big cell in the center is a glial cell, like a nerve cell. And you can see the Tregs kind of homing in, and as I like to say, scientifically technical term, glomming on to that cell. They're basically localizing to the cell and providing a shield of autoimmune protection. Finally, I just want to close out by saying that we've really put a stake in the ground and committed to this program. We built, I think I can safely say, the world's largest Treg manufacturing facility. It's a building in a building, much like our cubicles upstairs. The pictures on the right are scaffolding on top of this facility. There's 15 of those purple doors you see there. Each one can process 2 patient samples simultaneously, so we can do 30 at a time, 1,500 programs a year. We've been doing manufacturing for 17 years. I'll show you in a minute, we do have additional flexible expansion space. We can also produce, though, CAR-T, TCR-Ts, TILs. We're looking to do this with partners, so we could actually work with you independent of autoimmune and be a provider of manufacturing for Europe. That's the expansion space, so we haven't built out everything yet in this facility. And then finally, where we are is we are, after the phase 2 in autoimmune, we have EMA agreement that we can move forward into a registrational study. So within 2 years, we could have the first Treg approved cell therapy. Again, some of the other competitors, it's going to take 5 to 7 years before you get an approval. While we're generating revenue from that first round of approval, we can be expanding into other autoimmune diseases. It will be following on with the antigen-specific, the CAR, and the TCR Tregs. So let me stop there, and I thank you for your time and attention.